Hello, my name is Carol and I am a LICOR Solutions and Support Scientist. In this lesson, we're going to be talking about the best practices for your chemiluminescence Western blot when it comes to everything that happens between rehydration after transfer all the way down to the blocking step. Now, if you're anything like me, you may have been questioning yourself if the blocking step is even necessary. I mean, the primary antibody is going to be diluted in blocking buffer anyway, right? Some other questions that I hear a lot are, should I be washing my membrane after the blocking step? Or is it okay to keep my blocking overnight? So in this lesson, we're going to be talking about all of that and much more. The Western blot protocol can be divided into 3 parts. In part 1, we have to extract the proteins and estimate their concentration so that we can then separate them by molecular weight on a polyacrylamide gel with the aid of an electric current. In part 2, we transfer those proteins from the gel onto a membrane, again with the aid of electric current. In part 3, we will be probing for specific targets of interest with the aid of primary and secondary antibodies. And that's exactly the step we're in right now. So we're about to start part 3 of a chemiluminescent Western blot protocol. Before you start this part, just like with any other protocol, make sure you fully understand the protocol and that you have all of the reagents you need. Last but not least, remember to make sure you're always protecting the membrane from dust. And I do mean it at all times. Make sure those incubation boxes are always closed. Throughout the entire Western blot protocol, we want to do everything in our power to ensure the proteins remain on the membrane. Now, that may seem obvious and maybe even simple, but there's a lot of little things that can lead to protein detachment and you may not be aware of. For example, did you know that if you place your membrane protein sign down on your incubation box, those proteins will rub on the incubation box and that could lead to protein detachment? Same thing happens if I have more than one membrane in an incubation box and those proteins rub on the other membrane. Because of that, we recommend that you always have only one membrane per incubation box, protein side up. And just to elaborate on that, if you cut your membrane in half, each one of those halves is now considered a separate membrane, 2 different incubation boxes. Now, if we end up having protein detaching from the membrane, we're going to have empty areas. And those empty areas will compromise protein quantification. So we really want to avoid that. Another cool little trick to try and avoid protein detachment from the membrane is to never pour any solution directly on top of the membrane. You always want to tilt your incubation box and then gently pour the next solution onto the wall of that incubation box. We want to preserve the 3-dimensional structure of the membrane. If the structure of the membrane is damaged, proteins may detach from it, creating empty areas that will compromise data quantification. Similarly, if the structure of the membrane is damaged, background may be generated on these areas. And again, that's something that we definitely want to avoid as it can compromise data analysis. Because of that, you never want to place anything that is heavier than filter paper on top of the membrane. That includes holding your membrane. Don't touch your membrane directly. If you need to move it, use a pair of clean forceps and gently grab it to the edge of the membrane. One more reason why we don't touch the membrane in addition to try and avoid disrupting its structure is the fact that our hands and even our gloves have oils that could be a source of background. To ensure an even staining and low background, you always want to make sure you're using enough volume to fully cover the membrane in all of the steps of this protocol. It's also very important to keep time and temperature consistent. Using fresh reagents is also going to help you control for different variables and have accurate data. Another cool little trick is to always place your incubation box on a shaker with a gentle cycle so that the solutions are always evenly covering that membrane. Now, it is super important to keep your buffering system consistent. As you have seen in the science section with Gretchen, PBS is not compatible with the detection of post-translational modifications such as phosphorylation. The reason for that is that the primary antibody may bind to the phospho present in the buffer, which will compete with finding the actual antigen of interest, compromising data accuracy. Also, PBS is not compatible with detection systems that depend on alkaline phosphatases. The reason for that is that the phospho in the buffer may inactivate that enzyme. So remember, once you have determined which buffering system you're going to use, keep that consistent throughout the protocol and throughout your different experiments. Last but not least, remember to work in a clean environment. We want to clean the incubation boxes and the forceps to make sure we don't have any unwanted source of background. To that end, you can clean them with soap and water, rinse it very well, and then a quick rinse with a little bit of pure methanol. Remember, however, to not leave any residual soap or methanol, since that could interfere with the protocol and affect your final outcome. At this point, we're ready for our next step, and that's what we're going to be talking about on our next lesson. What are the best practices on how to rehydrate your membrane after transfer?